9 years, maybe almost 10 years. I worked in multiple labs across 2 universities where I did my master's and my PhD, and I thought it might be an interesting idea to show you what a typical molecular research lab looks like, or the kind of equipment and tools that you can find inside. I'm not going to give you a tour of the lab per se, this lab, but I will just show you some of the common equipment that you can find in a research lab that focuses on molecular research. One of the most common tools that you will find in a research lab are these tools called micropipettes. So as you can see, we have different sizes which allow us to manipulate different volumes of liquid. So here you can see we have two which can transfer between 0.2 to 2 microliters, 1 to 10 microliters, and then these two yellow ones on the left and the right between 10 to 100 and 20 to 200 microliters, and then the big one in the middle which can transfer between 100 to 1000 microliters. So we use these together with these kind of micropipette tips. So you just attach it like so, and then we use it to like transfer liquids from larger containers to smaller containers, or from smaller containers to other smaller containers. These little plastic tubes are what we often use together with our micropipettes. So I've heard them called microcentrifuge tubes or micropipette tubes. They basically can hold up to 1.5 milliliters of liquid. So we use these to mix liquids together for experimental use. We can also use them for storage in long-term storage like -20 or -80 degrees Celsius. This right here is a centrifuge. So this is basically a very fast merry-go-round for liquid samples. So usually we would like put our liquid samples in those little tubes that I showed you earlier, put them in here, and then they would spin around really fast. So this is usually done to separate solid particles from our samples if you want to have a clean liquid sample with no physical particulates. And these centrifuges come in multiple sizes. So here's another one. So you put all your tubes in there, and the one that I use the most is this one, because this one can take larger tubes like this, or the blood collection tubes. And the main reason I used it is to separate blood into its components. So we would take whole blood samples, put them in here, spin them around, and then we would get 3 layers inside the tube. Bottom layer being red blood cells, top layer being plasma or serum, and in between we would get like a thin layer of white blood cells. Sometimes the molecular work we want to do requires a sterile environment to avoid contamination of our biological samples, and so we would do it in this machine, which is known as a laminar flow cabinet. So the inside of this cabinet is a sterile environment. We can turn on a UV light to sterilize the inside and all the tools that we want to be sterilized, and when we turn it on, it will activate a blower which will circulate air in a way that the inside will only receive clean air from a filter that's at the top. So most molecular work can be done just outside on a normal bench, but sometimes when you need a sterile work environment, that's where this laminar flow cabinet comes in. These right here are technically called thermal cyclers, but we also just call them a PCR machine. So PCR stands for polymerase chain reaction, and it is basically a technique that we use to amplify DNA. But instead of merely amplifying an entire genomic sequence, PCR allows us to amplify specific DNA sequences that we are interested in. So one of the most recent and probably popular, well-known applications of PCR recently is COVID-19 detection. So as we know what the genetic makeup of the COVID virus is, we are able to amplify DNA sequences taken from saliva samples or nasal samples, amplify the DNA, and if the COVID DNA is present in the sample, these machines will amplify it, allowing us to detect whether or not that sample has COVID inside. An extension of PCR, which is what I used for my PhD, is real-time PCR. So similar to the basic PCR, this real-time PCR allows us to amplify specific genetic sequences, but while PCR only allows you to determine whether or not a DNA sequence is present, real-time PCR allows you to actually quantify the amount of DNA present. So we usually use real-time PCR for measuring gene expression. So if you want to see how much a particular gene is expressed in a sample, we can do real-time PCR to quantify it, and we can also use it to quantify bacterial or viral loads. So for example, using COVID again, if you wanted to know if an individual had a high or a low COVID burden, you would use real-time PCR to quantify the amount of genetic material belonging to the virus. This right here is another very important piece of equipment. It is called an autoclave, and it basically sterilizes any equipment, reagents, or stuff we need to use in the lab. So it sterilizes it through the use of steam, and as you can see here, somebody has already sterilized their equipment. It's currently at 83 degrees Celsius, but during the sterilization stage, it gets up to 121 degrees Celsius, so very hot steam. And so this is a very important piece of equipment for the lab, especially if we are dealing with samples that need to be conducted with sterile equipment. The final piece of equipment I want to show you is this, which we call either a spectrophotometer or a microplate reader, which we can use to quantify proteins in a sample or any other kind of compounds that we want to measure. So what we would do is we would take these little culture plates or micro well plates. We would insert samples into each of these wells, mix those samples together with enzymes and substrates, and then the samples will change color. And depending on the concentration of the protein that we are measuring, for example, the intensity of that color will change. And so what this machine does is it measures the absorbance in each of these wells, and it will tell us how much of our target protein, of our target compound, is in the sample. So this machine will allow us to compare the concentrations of different molecular targets in different samples. I use this probably the most throughout both my master's and my PhD. So that's it for this video. There's a lot more I didn't show you, like the cell culture labs or the equipment that we use to run gel electrophoresis, which we can use to separate bands of proteins or separate bands of DNA. But that's alright, I just wanted to show you some of the basic equipment that I'm quite familiar with and that I use throughout my master's and my PhD. Maybe I'll do another one of these videos to show you other lab apparatus that I can find around our lab here in the faculty. So yeah, that will be it for this video. Thanks for watching.